Okay, in this video we're going to have a look at the structure of proteins. And we know about proteins. We know that they're in meat and eggs and milk, and we know about them in the food. What we're going to have a look at is the structure of these molecules, and these are really interesting looking molecules, all with really specific shapes. They are organic molecules that are produced by organisms, and they're also polymers. Okay, the basic unit of these is an amino acid. And let's talk about the structure of these amino acids first. Amino acids basically have a carboxyl group with that carbon and the oxygen, an amino group with the nitrogens and the hydrogens, and they have an R group, and that is a variable group that makes it different. There's actually 20 different types of amino acids, and each one of them has a different R group. When we construct the proteins, we put these amino acids, and these amino acids are shown here. The different types of amino acids are shown here with these 3-letter codes. And these amino acids are the monomers that are making up our polymer, which is the protein. And these amino acids are joined with a peptide bond. And if there's 2 of them, it would be a dipeptide. Because we have lots of amino acids, we call it a polypeptide. Now, the structure of a protein is a lot more complex than just that. We have the primary structure of the protein being that order that we just looked at, the order of amino acids, the sequence of amino acids. And you can imagine with 20 different amino acids, you could come up with a whole lot of different combinations of that primary structure. The next thing we do is we either we can organise these strands of amino acids, and we can organise them in something called beta-pleated sheets or alpha helixes. And both of these structures get the shape, the beta-pleated sheets or the alpha spirals or helices have their shape because of the hydrogen bonds between the strands holding them in this organisation. And we call that folding and curling or coiling of the strands the secondary structure. The next level is a tertiary structure, and we end up with some complex bonds, some hydrogen bonds, some disulfide bonds between different areas of the sheets and the spirals to give a really complex tertiary shape. And that complex twisting and turning is what gives its really specific 3-dimensional shape. And the final structure is a quaternary structure. And that's where we're actually getting a few of these different polypeptides and joining them together, putting a few different puzzles together and making something a bit more complex. So that's the 4 levels: the primary, secondary, tertiary, and quaternary structure. And we can make all sorts of different shapes. We've got some examples here of some models of some of the shapes of these proteins. And that specific shape is what allows proteins to do things. So these proteins are the things like enzymes, or the types of proteins that are on the surface of cells to help with recognition. And we sometimes refer to that as having shape specificity. A shape that fits. And that's very important with signalling, with recognition, and with just molecules fitting together in things like enzymes. Okay, good luck with studying proteins. I think it's a really important organic molecule and macromolecule to understand. It'll creep up lots of times through the unit, from signalling molecules to diseases, to recognition on membranes, in enzymes. They'll just keep creeping up all the way through the year. So it's really important to learn them when we can. The really important feature of them is that shape specificity. Okay, good luck.